Hello, everyone. My name is Rupa Paik. I am the Head of Technical Operations for Cell Therapy at Thermo Fisher Scientific. I'm really excited to be part of this 5 days of stem cell event. Today, I will be speaking to you about the challenges in transitioning from discovery and development to GMP manufacturing with the goal of providing some practical guidance to make this process as seamless as possible and to achieve success. In the past 2 years, cell therapies such as CAR T therapies have achieved great commercial success, and as a result, cell therapy developers from academic centers, small startups, and large biopharmas continue to invest a lot of efforts in developing treatments for unmet medical needs. The ultimate goal to manufacture products is that they are safe, efficacious, scalable, and yet cost-effective. Many of you are familiar with cell and gene therapy. Gene therapy involves alteration or supplementation of a gene to resolve a genetic abnormality. Cell therapy, on the other hand, uses living cells to replace damaged or diseased cells. Cell therapies can be classified into autologous and allogenic. They can also be classified as gene-modified or non-gene modified based on the manipulations required to alter or enhance the functional properties of the cell. This slide summarizes the recent commercial success in bringing both cell and gene therapy products to market. In August 2017, FDA approved Kymriah, developed by Novartis for the treatment of ALL. Soon after that, in October, FDA approval came for the use of Yescarta, developed by Kite Gilead for the treatment of diffuse large B-cell lymphoma. In the gene therapy arena, 3 products were developed and approved between 2017 and 2019. They are Luxturna, Zolgensma, and Zynteglo. This slide speaks to the exciting landscape for regenerative medicine-based clinical trials, which of course include both cell and gene therapy trials. This information is from Alliance for Regenerative Medicine website, and as you can see, when this data was put together, there were a total of 1,069 ongoing clinical trials. 659 of those are for cell therapy, and 366 are for gene therapies. If I can draw your attention to the bottom of the panel, you can see that a large number of these were CAR T and TCR type trials. There is also a large number of trials using mesenchymal stem cells or MSCs. So, for these trials to achieve clinical and ultimately commercial success, it is imperative that scientists developing these therapies start thinking about 2 things at a very early stage. Those are 1, de-risking the process, and 2, scaling up the process without compromising safety and efficacy. Many people refer to this as a clinical vision. So why is clinical vision important? A clinical mindset is critical for successful transition of cell therapies from bench to bedside, especially since, like Kymriah, many future cell therapies may have their origin in academic organizations, hospitals, or small startup companies. It is important for scientists developing such technologies to understand and appreciate the transformation journey that their R&D observations need to go through to ultimately become a safe and effective therapy. So we have to start addressing the uniqueness of cells as products and the challenges associated with such a journey. Cell therapy manufacturing is unique. With the growing number of cellular therapies at various stages of clinical trials, it has become increasingly evident that cell therapy manufacturing is far more complex compared to conventional pharmaceutical manufacturing. In case of cell therapies, the process is the product, and therefore cells become what we call living pharmaceuticals. They are biological products that are highly sensitive to their environment and therefore subject to change in their attributes with the slightest of modifications in the parameters that control them. Autologous and allogenic processes may require completely different strategies. All of this to say that in cell therapy, there are no one-size-fits-all type solutions for manufacturing. Constantly evolving regulatory challenges also add another layer of complexity. This slide speaks about some overarching challenges associated with manufacturing of cell therapies. Need to manufacture consistently safe and effective product from highly variable starting material is a challenge. This could be apheresis, mobilized and non-mobilized bone marrow, or PBMCs. Cost of raw materials adds more complexities to that. Lack of robust, scalable, industrialized manufacturing, for example, maybe lack of automation, outdated analytics, and diverse manufacturing platforms currently on the market that lack standardization is another example. Chain of custody and chain of identity issues, which are unique to the entire process, because this process begins with patient or donor material and ends with getting the cells back into the patient, is also an important factor. Shortage of well-trained workforce is something that we all are facing, and of course, the high cost. As we all know, a single dose of Kymriah is over $400,000. So, what does it take to go from bench to bedside? In order to successfully transition from bench to bedside, it is vital to understand the essential steps for optimal manufacturing. Many manufacturing areas, including pharmaceuticals, apply the 5ME principles to gain efficiency. This is a comprehensive analytical approach to manufacturing, and it results in higher productivity, improved quality, and lower working capital. If you apply the 5ME principle to cell therapy, efficient manufacturing would integrate what is shown on this slide. Raw materials, which also includes the starting cellular material, methods and instruments used for manufacturing. This could be platforms, partially automated, fully automated, use of closed bags and tubing, and any of these kinds of instruments and platforms. Metrics would be the quality principles that you apply to your manufacturing process. And man, which actually refers to trained personnel. In the next few slides, my goal is to walk you through each of these with the intent of providing some practical steps to achieve success. So raw or ancillary materials. Raw or ancillary materials are defined as components used during the manufacturing process. They exert an effect on cell product, but are not intended to be part of the final cell product. They are important because they can impact safety, purity, and potency of the final product. Choosing the right raw materials will ensure an easy transition from basic research to clinical application. We need to evaluate the quality of raw materials. It is best to use the highest grade of materials that have well-defined components, especially for media, cytokines, and supplements as they affect the health and the properties of the cell. Whenever possible, it is best to use GMP-grade materials rather than RUO materials. We understand that GMP-grade materials are expensive, so at least incorporating them in the process development stage is important because process changes made due to raw material issues will present risks, require further comparability studies, and result in higher costs or delays. We need to choose the right supplier too for these raw materials because we need the suppliers that we choose to be able to provide the materials for all phases of the process, all the way from clinical to commercialization. Also, it is a good idea to have a second or a backup supplier for critical materials in case there are issues with the first supplier. And lastly, it is never too early to conduct some type of exercise to determine the cost of goods, because as the process scales up, this cost may be prohibitive to commercialization. Another unique aspect of cell therapy is the nature of the starting materials. The starting materials for cell therapy are cells. They are referred to as cellular raw materials or CRMs. Since donors and patients are unique and diverse, there is an inherent variability and heterogeneity in these cell populations. Treatment histories and comorbidities faced by the patients can also contribute to this variability. Healthy donor cells are used to generate R&D enabling data, but as we all know, healthy donors can never be true surrogates for patients. So what are some of the steps that we can take to achieve process efficiency at an early stage? Process efficiency needs to be achieved in such a way that changes to the process maintain product safety and they do not impact product quality, complexity, cost, or time. The process is what defines the final product because in case of cell therapies, the product can never be fully characterized. So we can achieve process efficiency by a few of these points. One, by conducting risk assessment to identify open steps and see if they can be closed using bags and tubings instead of conical tubes and traditional culture vessels. Closing specific steps will definitely reduce errors and chances of contamination. Two, any changes made to improve processes need to be validated with respect to time, raw materials, and money. Thirdly, considering equipment modifications or automation so cells remain in closed systems as much as possible. Fourth, conducting experiments early on in the game to address the scalability issues and make sure that if the process is scalable, it still maintains quality and efficacy. Fifth, to devise test methods or analytical methods that help assess the manufacturing process and ensure that these methods are fast and not labor-intensive or expensive. And finally, it is important to develop superior record-keeping skills and get familiar with data management, SOPs, and technical report type of documents. This slide shows an example of process development. As you can see, there are a variety of steps in this process, starting on top with cell isolation all the way to cell storage. Focus on process efficiency will enable the scientists to not just incorporate, but also evaluate all the steps and ensure that cells in each and every step still maintain safety, reproducibility, consistency, robustness, and sterility. I want to focus a little bit on technology transfer because many people believe that introducing technology transfer at an early stage will lead to smoother transition between R&D and PD and eventually PD and GMP. I want to talk a little bit about technology transfer. Technology transfer is a formalized process for moving the manufacturing process from process development teams or PD teams to the manufacturing team. Technology transfer creates a bridge between R&D and PD and helps towards successful GMP execution. Development, transfer, production, and documentation are the phases of successful technology transfer. And this involves knowledge transfer of the manufacturing process, both scientific and technical, information and training for the use of specialized equipment, complete list of raw materials, including all certifications and documents, training of personnel, and finally, development of technology transfer documents and all the related SOPs. I want to speak a little bit about analytical methods. Analytical methods are critical as they control the manufacturing process, raw material intermediates, and the final product. In cell therapy, analytical methods focus on product attributes such as identity, purity, potency, safety, and stability. Identity describes cell surface phenotypes associated with the therapeutic cell of interest. An example for identity would be flow cytometry measurements for the expression of chimeric antigen receptor for CAR T cell. Purity. Examples of purity would include cell surface marker expression associated with both unwanted cells and cells of interest, and it's typically measured by flow cytometry or qPCR. Potency is asking the question, are cells as a final product capable of performing the desired function? And examples would be interferon gamma secretion or cytotoxic assays for antigen-specific T cells. For safety, compendial tests are typically used. But in cell therapy, there are a lot of efforts to develop surrogate assays for a quick turnaround time, especially for fresh products. Typically, safety tests include endotoxin, mycoplasma, rapid mycoplasma, and sterility testing such as the 28-day, 14-day, or even a 4-day BACT test. Stability focuses on one thing. Does long-term cryopreservation of the cell product affect the quality of cells in terms of viability and potency once the cells are thawed to be infused or delivered to the patient? So as you can see, development of sound analytical methods is critical for the long-term success of cell therapy. I want to talk a little bit about quality by design or QBD because this is not an R&D concept, but many people believe that R&D teams should have some understanding of the QBD principles. Conceptually, QBD is simple. As a manufacturer, you want to design consistency and reliability into each step of the manufacturing process. The biological complexity of cells in cell therapy poses many challenges to the manufacturing process. The solution to these challenges is to design cell manufacturing process according to QBD principles. One can apply QBD principles by 1, linking measurable cell attributes to the final product quality, monitoring process changes during the manufacturing process to ensure the desired effect on cells, conducting risk assessment and using design of experiment principles to design sound and reproducible experiments, building strong quality management system, QMS, that captures written procedures and changes. QMS is not possible typically at R&D stage, but it can be incorporated at the PD stage. And finally, an understanding of the target product profile. So we have talked a lot about the product, but what about the people who are responsible for making this product? On this slide, I want to talk about the GMP mindset that will help the R&D and PD teams to be successful. So what does it mean for the R&D and the PD scientists to have a GMP mindset, a mindset that will help them with two principles that are critical for success. One, early incorporation of some GMP type of rigor and discipline, and two, to be thinking about de-risking every step of the manufacturing process to the best of their ability. Some of the practical things that would help such teams are understanding of the GMP principles, knowledge and exposure to the challenges of actually working in an ISO 7 GMP facility. As we all know, this requires gowning for long hours, working in pairs with one person as an operator and one person as a verifier, and just getting used to a very stringent and controlled environment. A good understanding and appreciation of closed and automated systems is valuable. Ability to translate research-based processes into scalable processes is also critical. Skills for foreseeing and de-risking specific steps of the manufacturing process is also very valuable. For example, when the cells need to be centrifuged, can we use bags instead of tubes for centrifugation process? Those type of things are very useful and critical in de-risking the process. And lastly, excellent record-keeping skills. So what are some of the practical ways of developing high-performing teams? Here are just a few ideas that I hope will be helpful. Tours of academic and commercial GMP facilities will be helpful. To take courses in clinical trial design, regulatory requirements, commercialization, and ethical research. Cross-training of cell biologists and engineers. Training to understand quality assurance and quality control, and also, if possible, training in data integrity, record keeping, and document management. So in conclusion, developing an effective, reproducible, and GMP-compliant manufacturing process is the foundation for successful manufacturing of cell therapy products. We can achieve this by developing a clinical vision at a very early stage, choosing raw materials carefully as we develop the process in R&D labs, de-risking the development process as we build it and transition it from R&D to PD, implementing robust and reproducible analytical methods, applying QPD principles whenever possible, and lastly, developing teams with a GMP mindset. Long-term success for cell therapies will come from adopting a systematic, phase-appropriate strategy, continuously thinking about de-risking the manufacturing process, and keeping in mind the end goal, which is, am I taking the cells through a manufacturing journey that will keep them safe and make them effective when they reach their final destination? Thank you for your time.